Hello, my name is Kenji Yo. I'm a biomedical engineering major with a minor in biochemistry, and this is my honors capstone project, determining SOX10's role in inducing potency in irradiated salivary gland cells. So, patients with head and neck cancer undergo radiation therapy to treat it. Unfortunately, this therapy isn't precise, as a result, irradiating the salivary glands. This destroys the cells, leading to hyposalivation, also known as xerostomia. This leads to disrupted oral microenvironment, dental caries, taste loss, oral infections, and difficulty in swallowing. Furthermore, the salivary glands can't repair themselves as there aren't enough stem cells, since those are also destroyed by radiation, as the stem cells are responsible for being a source of cells to replace the damaged tissue. Thus, our lab has been trying to develop a stem cell transplantation technique to address this. So previously, we have proven that injecting stem cells expressing the KIT receptor leads to repair and restoration of the function of the salivary gland. In further investigations, we found that SOX10, a transcription factor, is key to the proliferation and differentiation of the KIT-positive stem cells. Transcription factors are proteins that regulate gene expression, controlling the behavior of the cells. So we induced SOX10 expression in vitro and saw that it makes cells exhibit a stem cell-like characteristic. And now we want to replicate the clinical setting and do it in irradiated cells. And thus we hypothesize that SOX10 promotes potency in both radiated and non-irradiated salivary epithelial cells. Now there are two assays to determine this. The first is a self-renewal assay, where we irradiate the cells and then culture them, and see how easily they grow and proliferate by examining the cell count. The second method is a differentiation assay, where we irradiate the cells and culture them in a special media that contains chemical signals that tell the cell to specialize into a salivary gland epithelial cell. In both, we induce SOX10 overexpression by transfecting the cells with a lentivirus, similar to how HIV works, that inserts a gene designed to promote SOX10 expression. Now, Figure 1 confirms the success of this transfection. DHA and SOX10HA is just a tag that we use to help identify the SOX10 expression that arises as a result of this transfection. Now, if you look at the bar graph, this is the qPCR results. We see that in the transfected cells, there's a 25-fold increase in the SOX10 expression. The image on the right also shows the presence of SOX10 in green. And as we can see that with the presence of green, this is good. And RFP stands for red fluorescence protein, which shows where the cells are. Ideally, we want SOX10 expression where the cells are also at, so we ideally like to see red over green. And now that we know that cells are overexpressing SOX10, let's move on to the assays. So for the differentiation assay, we see that the SOX10 overexpression in the irradiated stem cells leads to the formation of AQP-positive acinar cells. So the acinar cells are part of the salivary gland, and AQP stands for aquaporin channels, which are channels used by the cells to transport liquids like water and saliva, and thus is critical for salivary gland cells. And the presence of these channels show that the cell is specializing into a salivary gland cell. In both images, the cell is radiated, and we can see more green in the cells with SOX10 overexpression. We see more evidence in Figure 3 where we looked at three proteins, K19, SMA, and CD166. K19 represents ductal epithelial cells, which form the structures in the salivary gland. SMA represents smooth muscle actin, which are muscle fibers responsible for moving the secretions in the salivary gland. And CD166 is a marker for mucus-producing cells, which produce saliva. And we can see that in that image is that with SOX10 overexpression, we have a stronger fluorescence signal than without SOX10. And all of these markers indicate a differentiated cell, and thus prove that SOX10 can improve differentiation potential in an irradiated cell. Furthermore, in Figure 3, we looked at cell count for the self-renewal assays, and you can see in the bar graph that SOX10 also promotes and restores cell count to almost control levels, thus preserving the self-renewal capacity of the cells. However, you may have noticed that there is a lack of quantitative data, specifically Western blot data, indicating the presence of proteins, as well as more qPCR data for three proteins that we mentioned before, as well as SOX10 expression in these cases. This was due to the COVID-19 pandemic, as due to the project timeline, we had planned on obtaining almost all the quantitative data in late March/early April, but unfortunately that was when the lockdown started. Fortunately, the data is promising, and so far it proves a hypothesis, but we still need more evidence for a stronger case, and we expect that the qPCR and Western blot data to align with this given what we have so far. Afterwards, we had planned on continuing the same experiment in mice and possibly exploring the effects of certain biomaterials as scaffolds to assist in maintaining cell viability in a stem cell transplantation procedure. Yes, this is my honors capstones, and if you have any questions, you can go down to the comments below. Thank you.